our panel here. So we've got Dr. Amy MacDonald and Dr. Dan Lumsden, who we've heard from this morning. Also, we have Professor Santosh, who will be talking to us later, I believe, and Dr. Shetty from Edinburgh, again, another paediatric neurologist who will be talking to us later, and then Jim Selfridge on the end and nearest to me. So I've got questions. I've tried to categorise the ones that I've got into sort of themes as we go through, and I suspect that there'll be some that are obvious for particular people to answer, but I'm not going to direct them to any individual panel members unless no one puts their hand up to answer. So we've just got a few questions first off about Rett syndrome and its genetics itself. We heard from Dr. Lumsden that a number of people have changes in their MECP2 genes but actually don't have a clinical syndrome of Rett. And a couple of questions have been asked as to why this is. Why don't all people who have the MECP2 gene changes present with Rett syndrome? Is it specific changes in the MECP2 genes that cause the specific Rett symptoms? I don't know who would like to.
Yeah, I mean, that's actually a very good question. What it reflects, and both Daniel and Jackie touched upon it, MECP2 is actually quite a complex gene. It's quite a large molecule. But what we've learned over the years, where there's these two key elements, the MBD, the bit that binds to DNA, and the NID, the bit that recruits the other molecules. They're the key functional domains. And what we also showed within Adrian's lab is pretty much you can take away the rest of MECP2 as long as you've got your functional MBD and your NID. So we actually made a mouse with that version of MECP2, just the MBD and the NID, and the mouse was largely unaffected. So what the key difference is if you have mutations in those two domains, largely speaking, then you will become affected by Rett syndrome. Most of us in the room today will have a different MECP2 sequence because we're all different. But most of us in the room won't have the differences in the MBD or the NID, and that's why we're unaffected. Those that manifest Rett syndrome will typically have these genetic changes in either the MBD or the NID. There are other mutations out with those two domains that also cause Rett syndrome through a different mechanism. So the truncations, where your MECP2 molecule might be a little bit shorter, and what that does is it means you haven't got the NID, and that's where you end up with Rett syndrome. So it's these sort of genetic variations in the general population that explain the differences and why some people will have a different MECP2 sequence but do not have Rett syndrome. I don't know if that was clear enough. If not, please speak out. I'm a scientist, and yeah, I'm not used to speaking to people. We get lots of learning on that. So yeah, this is a great day for me. I'm getting the chance to speak to you guys about something I care about. So yeah, please reach out and ask me any questions you want over lunch or whatever.
Thank you. As someone who's not used to speaking to people, you've done very well there, so thank you. And there was another question relating to whether there's correlation between mutation type and severity of symptoms. And I think one of your slides, Dan, showed that there's a spectrum of some correlation between the mutation type and the severity of symptoms, but it's not specific enough that as clinicians, when we see someone, they show us their gene change result, that we can say definitely you are going to fall into perhaps the more severe or the less severe category, because there's different amounts of expression depending on the inactivation of the X chromosomes in the cells, which is random. So someone may have a lot of their good X chromosomes inactivated, and so they might be more severely presenting, or you may get the converse where lots of your affected X chromosomes are inactivated, and so you have less of the sort of faulty expression, as it were. So there's similar to Rett probably being a spectrum, there's probably a spectrum of changes that we see there. An interesting question. So I think thinking about the fact that a lot of people affected with Rett syndrome perhaps have progressive slowing of the head as they get older. Is the brain growth, is that limited or affected by the MECP2 activity? And if so, and this may be we may need to come to you, Jackie, possibly, did the brains of the mice grow more once they'd been treated? Was that something that was looked at?
It was. There was a follow-up study where we looked in more detail at various aspects of the brain. We found that the neurons actually got bigger after the reversal. Before they were smaller than normal. I'm just trying to remember the details. Measured cortical thickness, and I can't remember if that was reversed.
Slightly.
Yeah, I think it was. Yeah, and the complexity of dendrites and neurons also increased after treatment. So the brain does kind of grow back to normal. As a non-neuroscientist.
Can I move out to that? Is that okay?
Yeah, absolutely.
Perfect. So I think, I'm terrible with the volume level, sorry. So I think the important thing is you don't lose neurons. So this is not a disorder where cells are lost or cells are damaged, and that's hugely important because brain cells don't really regenerate. So the liver, you can, and I'm not advocating going out and giving your liver a kind of kicking tonight, but the liver, other organs can really regenerate a lot. The brain cells really, there's a very limited capacity for any regrowth there. So the cells are there, but they're just not as complex as they should do. So when we talk about the faltering of the head growth, it's not that the brain shrinks or the head shrinks, it just doesn't grow as it should there. One of the big differences between the mouse and the human is the complexity of the cortex of the outside of the brain in the human model there. And again, this is why all of those things which are so important to us around communication and cognition, it's the cortex, the much more complicated surface of the brain that humans have compared to the mouse model there. But I think we are expecting and anticipating that there is some degree of recovery, and of course that creates the challenge of if you've got a small brain and a sealed container of the skull can't really grow very quickly, so might we get into a situation where if we reactivate some degree of brain growth and development, actually we start to get a bit of a build-up of pressure. So that's one of the things that we need to be monitoring for, and it's one of the reasons why there are repeated MRI scans on both of the trial protocols.
Can I just add a
Oh, straight.
I mean, the other thing to think about in clinical practice, it's quite important that the other complications of having a neurodisability over the years, for example, if you have had a status epilepticus, for example, and for those who have epilepsy, and that can cause a secondary brain injury. And although we are thinking that from a neuronal perspective this is good, but there are secondary effects of having a neurodisability. You could have a prolonged chest infection and you could be in the intensive care, or you could have long seizure and you could have had a secondary damage. We know, for example, a parallel is that somebody who has a febrile seizure, which is very common in children, and a small number of children who have febrile seizure can have a temporal lobe injury. And these are the effects you may not kind of gain back to the same degree in humans. It's really important to think about what other clinical impact the condition had over the years. That's an important aspect from a clinician's perspective.
I think one other aspect, exactly as Dr. Shetty said, is that even pre regression, there may be other factors like hypoxia or complications during pregnancy or delivery, because at least in our cohort of patients, we've started seeing that there may be accelerated regression arising in the group who actually have had pre-existing additional injury, which is on top of the MECP2. And so because we haven't been really looking at it, and the second thing is that similarly, we might end up seeing that there may be other rare diseases or mutations which are not just MECP2 related. So, for example, in a cohort of patients in our clinic, we've ended up seeing homocysteine pathway is also affected in around 17 out of the 65 kids. So suddenly, when you've got a double thing which is not directly related to MECP2, you may end up having very different trajectories which may not necessarily be related just to that one pathway.
Thank you. Hopefully that's clarified some of that. Anyone want to ask anything further? No? Okay. So another question relating to sort of the genetics of Rett itself. There were a couple of questions about this actually. So we heard about developing and breeding the chimera mice that were used for the models, and that these mice were then used to produce generations of affected mice. And this has raised questions for a few people about what they've been told when their child's diagnosed, that Rett syndrome genetically isn't inherited sort of through generations. And so does that mean that there is a potential in humans that it could be inherited genetically from people carrying the MECP2 gene change?
Technically speaking, it can be inherited because the gene was originally identified by Uda Zogby, where she had two girls presented in her clinic, so sisters, where they both presented with Rett syndrome, and that was extremely unusual. And the reason behind that was the mother was actually carrying the mutation. But as we've heard before, you switch off your X chromosomes randomly, and in her brain, the vast majority of the affected MECP2 copies had been switched off. So she presented with mild learning difficulties. So technically speaking, it could be inherited, and in those two initial patients, it was inherited from the mother. But typically, it's a spontaneous mutation that will just occur spontaneously in individual children. So that's why it's actually considered to be a rare disorder.
Thank you. And again, thinking about perhaps other factors that might affect the expression of the symptoms that we see in Rett syndrome in someone who's got a gene change. I think this is a question from a family whose daughter developed epilepsy at the age of 18 and was then diagnosed with Rett syndrome at the age of 29. And is it possible for Rett syndrome to lie dormant, or for other factors to affect why it might perhaps become more apparent over time?
Jay looked at me as if he wanted me to answer. Yeah. So I think it goes back to this idea of it being a spectrum of a disorder. So again, I think that idea of typical versus atypical is a little simplistic. We've certainly seen in our clinic young people who have not had an early regression, but at the age of kind of 10 or 12 have had a degree of regression at that stage, and then some more Rett-like features have emerged. And they kind of wouldn't fit into those kind of classical or kind of typical or atypical kind of boxes, but it's clear that it's the MECP2 gene and it's that range of that there. I think some diagnosis is unfortunately just late diagnosis, and it's just that people haven't kind of connected the dots and recognised the syndrome there. But I think the more we're seeing and the more able we are to do these genetic tests in young people and getting MECP2 results back in young people that we might not have thought about testing it for even 5 years ago, and certainly not 10 years ago, we're recognising it as a much broader spectrum of a disorder. But certainly I've seen, and I'd be interested in Professor Santosh's experience as well, young people with some learning disability or an autism picture who really don't look at the age of 7 like it is a Rett syndrome, and then something's changed at 10 or 11 and more features have emerged. Small numbers, I think it's unusual, but I think we do see that.
No, just to add a clinical, I mean, again, obviously, I'm not a scientist, so from a clinical experience point of view, we can draw parallel from other conditions, like kind of much more well-known condition for clinicians is Dravet syndrome, where we are probably very early onset, very severe form of epilepsy, but we are recognizing that there are gene mutations we found in adults with this type of condition. So you might have a much milder phenotype initially, and then one of these symptoms might come later on.
I mean, and we've also noticed quite a few of them like that, where in fact, sometimes when the changes occur and they assess the patient, you end up realizing that the original test for the MECP2 was actually outdated, and they now have a new genetic mutation identified as being the cause for it. So you can have very different reasons for it, but I think as Lumsden said, it's a spectrum, and that's where I think most conditions are going to end up being described as in the end.
Thank you. Okay, we're moving on to some questions about the actual sort of trials and gene therapy itself now. So there's been a couple of queries we've heard about the 2 delivery systems that might be used, so either intraventricular, where it's delivered straight into the ventricles of the brain, or through a lumbar puncture, given intrathecally that way. Would anyone like to comment on the safety profile of either approach, whether one is deemed to be preferable to the other, or particularly for individuals? Are there factors that might determine which would be more appropriate? I had Amy firmly on my mind for that question.
So hi everybody. So I'm not a Rett expert by any means, but I work in a lot of gene therapy clinical trials and delivering those trials to children with a various range of neurological problems. And I'm going to talk a little bit about delivery of gene therapy in my talk later on, but essentially the honest answer is I don't think any of us really know what the best way to give these gene therapies to the brain is. And so we do trials where we give it into the spinal fluid at the base of the spine. We do them where we give it into the spinal fluid at the base of the brain, and we do it straight into the ventricles as well. And some trials even put gene therapy into the actual brain tissue as well. And so we can talk a little bit about that later on. I think from a safety perspective, these procedures, we do them with our neurosurgical team primarily. So in some places in the US, sometimes that's a specialist interventional radiologist, but certainly in the UK, it's typically our neurosurgeons who do these procedures for lots of other reasons on a really regular basis. And so actually the safety profile of the procedure is really quite good. And so I don't ever have any anxieties about the delivery process. My anxieties are always more about the gene therapy product and all of the immunosuppression we'll talk about as well.
I think we don't know, but I think there's not a massive concern that we have. I think there's lots of reasons. Intrathecally is a little bit easier. So if I'm looking 5-10 years in the future, if we had a medicinal product that we knew worked, it will be a lot easier to set up surfaces that can deliver it through a lumbar puncture than it would be through the intraventricular route, just because the intraventricular route would need a neurosurgeon to be able to deliver that and a bit more apparatus there. On the other hand, lots of our young people have had scoliosis surgery, and actually delivering an intrathecal injection when there are rods in place is going to be very challenging. So I think there is a really good argument we can make for both of them, and that's why it's so fortuitous that we are getting the opportunity to test them both.
No, just from the family's perspective, just basically, you should be reassured that there is lots of experience delivering intrathecal treatment, either baclofen or antibiotics or various other things. So from a safety perspective, it should be fairly reassured. Obviously, there is no procedure without any risks, but it should be fairly...
Just fielding some questions from the room. How long would a potential stay in hospital be after administering either of these two therapies?
I mean, most clinical trial protocols usually require that a patient stays in the hospital physically for 3 days minimum. Some protocols are ever so slightly longer, and then usually there's a requirement that a patient stays within the vicinity of the hospital for the next following couple of weeks, usually, just so that we've got easy access to them if needed. But usually, I think the shortest I've done is a 3-day admission.
I concur. And I think hopefully when we get, again, for horizon scanning, when it's the future, when it's not when it's just the clinical delivery, I suspect that will be quite significantly reduced and maybe even just an overnight stay. But for the foreseeable future, it will be that admission.
Okay, I guess following on from that, thinking of some of the risks of the gene therapies themselves, people are obviously have heard of risks of, for example, cancers arising following gene therapies. Are there at the moment, or might there in the future be any modifications like DNA base editing or anything like that that would reduce the risk?
These questions are great. It shows how informed you are as a community. So yeah. So myself and Jackie have been working on Rett syndrome for 25 years, and clinical trials have started, but we haven't retired. And Jackie is actively doing experiments in the lab, looking at base editing. Had you asked this question 5 years ago, is base editing an option? I would have said no, because the technology wasn't good enough. But in that short 5 years, Jackie's now doing experiments similar to the videos you saw there, where she's doing base editing. And that is another option that could come along in the near future. We don't know. It's a technological advance, and how fast that goes, Jackie will be back soon to tell you about that. So yeah, we haven't given up. We're coming up with lots of different ideas, base editing. There'll be different AAVs. People are looking at that, see if we can supercharge the AAVs to make them more efficient, stuff like that. So yeah, there's going to be, it's still a work in progress, but it's exciting times.
Okay, and again, thinking about the risks of gene therapies themselves, we heard a bit about getting the balance between underdosing and so not getting a good enough effect, and overdosing and running into issues that way. A couple of questions about whether it's possible to individualise the doses in gene therapies to take into account those risks, or whether that's something that might become available.
Yeah, the honest answer is I think that's a really creative approach, and I think in the future we will, because we talk a lot about what we call precision medicine, so the right medicine at the right time for the right patient, and really personalising it, so personalising it to their genetic changes and personalising it to their own symptoms and time frame. And so I think in the long-term future, potentially, would be my answer. In the immediate future, we're really at the very first steps of safety, and the precision will come later, I think would be my honest answer.
Yeah. And I think in terms of that question of the balance, right, the data I'm seeing from both Neurogene and Tasha, it really reassures me that I really don't think that's going to be a problem. I can't obviously guarantee it, but I think it's really quite compelling safety data from the animal models and other models. I can't explain quite how it works because it's far too clever. I'm really a simple clinician for these things, but I think it's much less of a concern than, say, the immune response to the virus, which I think we do need to be cautious around.
Okay, and if all our safety measures and our experience put into place, and touch wood, this doesn't happen, if something dramatic or fatal happened with someone who was participating in these trials, what would happen next in terms of the process? So would the trial have to stop? Would it end the involvement of Taysha or Neurogene or whichever organisation in further developing any therapies?
So speaking as just a clinician, I think if there was a death at earlier stages of the trial would immediately halt to recruitment of new subjects, and obviously that would be scrutinised very, very carefully by the Independent Data Monitoring Committee. There's lots of unknowns around that, and I think it would depend upon the circumstances, why it had happened, and whether it was something that was directly attributable to the gene therapy or other comorbidities of the people that we're looking after. I suspect Amy will pick up on some of this from some of the other disease groups that she's got experience with, but certainly it would put things on hold for a period of time there. As to whether that derailed in the longer term any of the programs, that I think it's impossible to comment on at this stage.
Thank you. So certainly any safety event, any safety signal that, so maybe not even anything as significant as a death, would automatically pause, certainly in the very beginning stages, would pause study recruitment. Now that doesn't stop study recruitment, but it pauses it, and all of the data gets reviewed by an external committee to see whether we can understand where that safety signal has come from and what the implications of that should be. Sometimes at the very start of a clinical trial, you'll have staggered dosing for this exact reason. So we might recruit one patient, and then it might be 40 days before the next clinical team anywhere in the world is allowed to even think about enrolment. And there will be safety points along the trial which stops us from maybe escalating the dose or recruiting more patients or opening up to a specific subgroup. So it might be that you're open for children over 8 years, but you're not allowed to open for children under 8 years until you've met this safety marker. Any serious events all get reported and reviewed immediately. So if I have even a child admitted to hospital with something completely unrelated, that is regarded as a safety event that I have to report within 24 hours of that child being admitted. And again, that gets interrogated in detail. So we're trying to detect these concerns. The long-term implications, if there is something catastrophic that happens in a trial, it can lead to the premature stopping of a trial, which is good because that's safe and that's in patient interest. The long-term implication of future therapeutic development, it just become a scary place, and people will, a lot of our companies, and I don't know the circumstances with Taysha and Neurogene, but a lot of companies are dependent on financial support to maintain their space, and if there isn't the investment there, then they cannot continue their program. And we have scenarios where companies have lost funding and trials have stopped. And what we have to do as a community is to make sure that that doesn't label our disorder as untouchable. That gene therapy doesn't get labelled as we can't do gene therapy in this disorder anymore. And so that's the biggest risk I worry about for our rare diseases. But I think given that there is more than one commercial entity interested in disorder, we have labs around the world and we have multiple disease groups advocating for patients with Rett syndrome, I'm less worried about Rett syndrome than I am about some of my other super rare diseases. So I think the safety risks are present, they're taken incredibly seriously, and they can delay and slow development, but I'm not anxious for Rett syndrome specifically that it will stop all future development should something awful happen.
I'm not going to talk about safety, and then it's just more about practical implication for this, for the family, particularly at the early stages of safety trial. And I mean, I think it's one of the good things in NHS is the data sharing, and we will know from hospital admission, no matter where you are, but this still will have a significant impact on the families who are taking part in the early safety trial, staying closer to the hospital, because each of these safety data is very, very important. And even if you go to the local emergency department, the doctor's entry there, and from whatever treatment you receive. So these things have a very significant practical impact on how we manage an early safety data.
Thank you. Hopefully alleviating some worries about some of the safety aspects of the trials. Any follow-up questions from anyone about those?
Hi, my name is Lynn and I'm coming from Dublin with my daughter Jessica who has MEPC2. So we were just wondering, like, how do we get on to the trials because it's not done in Ireland? So like we don't mind travelling to the UK because we came today for it. So we just want to find out a bit more of how to get, you know, we have Jessica's genetics with us today, like everything to do with genetics. So if that will help today and whatever, thank you.
So again, I can't speak specifically for Tasha with the Neurogene trial at the moment, but for any other clinical trials, I would always, so a lot of centres will accept international referrals for clinical trials. And so certainly in Manchester, I would take a referral if it's part of the sponsors agreement, because we need them to fund travel, accommodation, all of the things that go with participating in a clinical trial. So it's very difficult for me to say for Neurogene and Tasha for these specific trials whether they would allow us to accept an international referral. But in routine practice, if anybody, you know, even if there was a trial that had opened only in the US, for example, as a clinician, I could refer you to that site for consideration for inclusion. And usually we'll show you probably later on the clinicaltrials.gov website, which hosts all of the clinical trials that are active, and it details which sites are open for recruitment, and there's usually a contact number. So families can self-refer for a lot of trials as well. And again, I'm going to kind of talk about that clinical trial journey during my talk later on. But yeah, I can't really comment for these specific trials, but usually you should be able to access any trial anywhere in the world.
I think also to just be really honest and open, these are phase 1 and 2 trials. The number of children and young people that are to be recruited by both companies worldwide, you can see that both companies are trying to open in multiple different territories. It's only going to be very small numbers of young people that are involved from each individual site at this stage. I think PI would be honest and say, I think it's very likely that it would just be children already known to us for the initial stage. But the hope is, of course, that if that safety signal is there's not a worry from that there, and things are to kind of expand, we would want this to be as open as we can. I've just been handed a bit of paper, which I think means I have to answer this. So why are they so few participants in the trial? So it's all around it being the phase 1 or phase 2. So this is all around demonstrating that it is safe, that we can give this without big problems, and then you design the much biggest trials that are then going to demonstrate that there's the efficacy to it there. So you have quite restrictive inclusion criteria to make sure that you're recruiting young people who are at a lower risk group of potentially having complications, because if you have very broad criteria, if you have children where there are lots of other health problems and comorbidities, if something happens which is always going to happen very sadly and is unrelated to the gene therapy, it's a big safety problem, safety signal, because it muddies that water there. So it's enormously frustrating for all of us that this is not something that we can rush to offer, but that's how it should be and how it has to be. And one of the reasons why I'm so grateful for Reverse Rett for bringing everyone together is my eyes on that longer goal of, well, how do we roll this out and make sure everyone is able to access it? The bottom line is that this is a rare disorder, but it's not that rare. And look around the room. You know, this is a tiny fraction of the young people that we all know and the families affected by Rett syndrome. We are not possibly going to be able to recruit everybody that wants to be on the trials for the UK, even if all of the recruitment for the world was in the UK. So what we need to be doing is making sure in parallel, if this works, if it's safe, whichever the gene therapy ends up kind of passing through the trial phases, we're able to roll this out on the NHS and in other health services there. So it's all of this. But we are not going to be able to recruit everybody who would like to be part of that. And I know how upsetting that is for us as PIs to turn people away. I can't begin to sort of imagine how difficult that is. But be reassured, the long game is to get this open to everyone, assuming it works and assuming it's safe.
Thanks. So picking up from that explanation as to why the numbers are so small and it's such a very restricted and specific group that we're starting with, as you probably anticipated, there's lots of questions about when either trials or is it likely that therapy will become available for people who don't fall into the very strict categories of typical Rett with specific gene changes. Questions about, would it be open to male people who are affected with Rett? And what the future looks like in terms of how broad we might expect this sort of therapy to become. So, take that one.
So a little while back I said there was 3 types of questions, and I think this unfortunately slightly falls into the third of we don't know. Not every clinician that is involved in this wants as many young people as we possibly can to benefit from the intervention. And one of the worst outcomes for me would be we go through all of this and we end up with a very restricted licensing, which means only a very small number of young people are able to benefit from it. And I say young people, we want this ultimately to be available to adults. That has to be the goal, it has to be. I am sure from each of the companies, they also want the indications to be as broad as they can, because let's be honest, that will bring in more pay, that will be more of a financial reward for it. And I think that's vulgar or cynical, that's just the reality of trials there. It has to start small, it then has to broaden out as much as we can, but all of us will be pushing to try and make it as broad as we can there. I think it's really difficult to know whether regulators will say, well, you've got good data for atypical, you've now got a true trial for typical Rett syndrome. And I don't know how the regulators are going to respond to that. Interestingly, trofinetide, which was the trials were all in typical Rett syndrome, the FDA has approved it just blanket MECP2 mutations. So that's an interest, and I hadn't realised that until this week speaking to the company there. But I think we just don't know how things are going to pan out. But all of us as clinicians want this to be as open as we can do. It may find out that there are groups that really benefit and groups that don't. We simply don't know that. It's just too early in the trial period. I'm going to hand over to Amy and
I would agree. I think part of what happens in the long term will depend on us as a community supporting the companies in their applications, so advocating for expanded access. But this again is so far away from where we're at right now, and I think we have to be really realistic and honest about that. We always start in a small cohort of patients and then we expand. Once we're into that realm, we can start to think about compassionate access, expanded use programs, and so those questions start to get answered. But we should be thinking that far ahead, because you should always be thinking 10 steps ahead, but we're not able to answer that with any honesty. And I think that's what we have to be. We have to be really honest with you and really transparent, because otherwise it's not fair. And I think there's a question here.
I am handing Amy a bit of paper.
Are adolescents and adult trials likely to be carried out in the UK in the future? I think it depends on the success of these current trials, in honesty. If there's a good signal of efficacy, if there's a good signal for safety, then I don't see why not.
I mean, just the parallel to be drawn again from the SMA, if the trials are all in children actually, and the adults SMA 1, 2, 3 are all getting treatment. So if we draw parallel from the other conditions, the other rare disease where if there is an efficacious treatment, I don't think you need to go through a trial again, particularly for similar mutations. So there are examples of not needing further trials.
Rachel's approaching menacingly with another bit of paper.
Is there an operation limit for tissue?
So the question is about for the Tasha studies and upper age limit. So it's 8 years of age, but that's up until the 9th birthday, is my understanding. I'm looking at Chelsea. I'm getting a Chelsea nod of approval, which is always lovely.
I'm sorry, it should be for adults.
For adults,
Yeah.
there's not an upper age limit for the adult trials in Canada, is there? It's 12 and above is the limit there. Yeah, it's a 12 plus.
That's all right. That's just that's rubbish.
Okay.
Thanks. So I think some of the questions that people have asked about the reasons why it takes such a long time from going from the stages of being in a trial process to a therapy actually becoming available on the NHS or through other means to people who weren't involved in the trial stages. I think a lot of that was answered by Dan's description of all the different processes that have to be gone through before a treatment can be approved and signed off, and then hopefully be made available to the wider population of affected people. But there's also lots of questions about looking at experience from perhaps other gene therapies. What's a reasonable or realistic timescale, if everything goes smoothly, to when a product may become more widely available?
Maybe I'll let you take that one.
I mean, if we look at some of the more recently approved gene therapies that are now funded, if we look at the gene therapies, we're looking at 10 years plus. If we're looking at simpler therapies, we are currently still fighting with NICE for an agreement on a drug that we know works beautifully for a disorder, but because we can't agree on price with them, that is limiting our access for therapy for kids with this other disorder. So it's a horrible system, and these are really expensive therapies. I think we're moving into an era where we're having so many more of these therapies so these systems have to change and they have to improve, and so I think we will see an acceleration. And there are systems in place, orphan drug systems in place to try and accelerate therapy to patients. But I don't sit here confident that we're anywhere close yet. So, I think 10 years is generous to say that we'll be in a position the way we are with SMA or with NLD, where we're telling you that you can be referred to a UK site for an approved NHS delivered gene therapy for Rett syndrome. But we have to start somewhere, and so we're starting.
I think it's fair to say as well there are other mechanisms before it's fully approved and funded. The managed access agreements, the compassion, so there are ways that we can access it. One of the things that we discussed at the meal last night is a big problem for us is we don't know the full landscape of Rett syndrome in the UK. So we go to NICE tomorrow and we say we've got a working gene therapy, and we say, okay, so how many people is this going to be applicable for? And at the moment, we can't give a very firm figure for that there. So this is a bit of work that we all collectively need to be doing now, working out where young people and adults are with Rett syndrome, which parts of the country, trying to get some data around their level of how severely they're affected by it. So we've got the complete package that we can to bring to the regulators to make the decision making as smooth and as quick as we can. So that's why there's lots of work that needs to be going on in the background and to get to that point. So it's that additional hurdle. It's the frustratingness that Emmy's mentioned around a drug that we might know works, but we can't easily give to people because we haven't got all of that information in place there.
I just only to add to that, actually, I think this is a good place where you have a very well organized community group and just basically expanding this. And then we're in Scotland, slightly different position than Sign and NICE, but again, from the health economics point of view, it's just gathering the data, as Dan says, about what would be the cost of not having a treatment in terms of the spinal surgery, intensive care admission, repeated hospital admission, the care, parents' employment, various other things. So it's for the community, for us to look at what is the cost of looking after a young person with Rett syndrome if we have an effective treatment. And that's basically much more easier if we have really robust data to say, here is the cost of looking after a young person with Rett syndrome, and here is the cost of the treatment, and here is the quality of life for the families. That's the kind of data which we need to start gathering now, as soon as possible. And then as a community, it's much more easier for us to go to the regulators to say, let's move this forward.
Okay, thinking about processes after someone's been given the gene therapy, there's some questions around what we might expect a successful or positive outcome of the gene therapy to look like, and what would need to be in place to support that. So would we anticipate that changes would happen just anyway as a result of having been given the gene therapy? Should it be thought of similar to rehab following a stroke? And also what psychological provisions have been thought about to support people who are being given the gene therapy as they go through those changes afterwards?
Really, really good questions. So we don't know the magnitude of the change we're going to see. And it's clear that any change you see to reduce an impairment and to unlock the potential for more function, you're going to have more therapy to be able to access that. So if you just reduce the kind of genetic impairment, but you don't then allow people to access more OT to use their hands, speech and language to access better communication, physio to access better mobility, you're not going to get the maximum out of the intervention. So my hope is that when we give these treatments, we have to send these young people back to their local teams, and their provision needs to quite dramatically change because their capacity and potential to benefit from that will change there. It's difficult to mandate that. I don't think you can standardise it because it should be individualised and necessarily so. But I think it's really difficult for us to know how much change we're going to see. I said before we went into it, I don't use the word cure. And I think I talk about shifting the phenotype, I talk about improving the situation. The phase 1 and phase 2 trial, it's not going to tell us how much improvement. It's not designed to do that. It's around, is it safe to go on to the bigger trials that are going to give us that information? And that is one of the big unknowns at the moment, we want to be honest around.
I mean, in terms of the psychological support needed for trials like this, I think it's quite significant, because if you just look at transplants as such, in terms of if there's a heart transplant, the whole amount of psychological support that is needed wraparound for a heart transplant is not small, because it requires a whole thing about a shift in the entire family's understanding of what is to be expected, what are the risks, what might need to change as a whole family in terms of where they live for the first 6 months because they need to be close to the hospital, or whatever it is. There is a whole lot of things. And the other thing is the ability for the family to be able to maintain the immunosuppression in the initial phase if it's needed. Because if that is not done, then the entire consequence of whatever the gene therapy would do could be negatively affected. So there are issues, and of course the other aspect of it is that sometimes when immunosuppression is used, you can have emotional and behavioral consequences. So of the immunosuppression, not the gene therapy. So that there is this whole issue of having to understand even if there is emergent, sort of new symptoms or side effects or whatever one wants to call it, the cause for it cannot be identified very easily because you may have a multi-level sort of causation in that. And then the other aspect of it is that I think probably if it is successful and you end up having especially cognitive or social understanding improved, then for someone who has been almost like a locked-in state, has not had the ability to experience that, would probably end up leading to quite significant emotional turmoil, which will require to be managed appropriately. So one of the problems which we raised yesterday and were discussing the thing was, At one level, actually, if it works, what might happen in the short term is that you may end up having to increase resources because none of these resources, so for example, a psychologist being involved in managing this care would not have been needed if therapy was not working. So actually factoring into it that if you're looking at a health economic evaluation, actually it might be that if it works, you may end up having to spend more rather than less because of what the potential for change is. And this is something that I have raised multiple times because it is not only in Rett syndrome, it's the same discussion has been had for other conditions where biologicals have been tried, and we've ended up in exactly the same scenario because, for example, enzyme replacement, and that has come with the cost of emotional consequences because of neurotransmitter changes, which then has meant that the need for neuropsychiatric input, which may have not been needed if that had not been used. But actually it was a positive in terms of all the other systems have got better. And so there is this sort of fine line between it. So I think even with the best scenario outcome, maybe one has to actually factor in that neuropsychiatric and psychological and other therapies will probably need to be enhanced, because otherwise we may end up wrongly concluding that things have got worse when actually it may not be.
This is a slightly different point, but it just some of the comments that we're talking about the psychological and emotional burden of a gene therapy intervention, and it just sort of sparked in my mind that it's really important that as a community that you support each other, especially in these really early phases. There could just be maybe one or two families in this room who get the opportunity to participate in these first trials. And that's going to be really tough on those other families who haven't had that opportunity. But at the same time, they're going to go on a journey which is going to be unique to them, and it's going to be really hard. And so they need your support, and you need to support each other. And so it's worth, I'm going to talk a bit later on about the burden of just being in a clinical trial full stop, let alone a gene therapy trial for an early phase experimental therapy in the most precious person in your life. But just be mindful of, it's really hard to think outside of your space right now, but what you're going through is going to impact everybody else in this room. And so you need to support each other as much as you possibly can, and as hard as that's going to be. So I think just be mindful of not just the psychological and emotional burden of the intervention, but of everything that goes with it.
And I think, just to add to that, it's all right not to take part in the trial. That's a really important thing. And I'm sure lots of you are sitting there thinking, well, I'd like this, but I'm really scared of it. It's okay. It really is okay. And you're not letting your trial down by not taking part in the study.
Thank you. I'm aware that we are coming very close to lunchtime now. I've just got one last question, which was one of the first ones that we received in, so I'd feel very bad if we didn't get opportunity for this to ask too. Again, a really good question, and again, I'd echo what was said before, that it just shows how much everyone is really invested in moving this process forward, really understanding it, understanding what we need to do to help make sure that that this is as successful as possible. So we've heard that these are the sort of first phases of trials that we're starting. Hopefully, not only will there be some successes, but there will also be information that helps us to understand how we can perhaps change, improve, modify the therapies that are offered. And a few people have asked, and again it was answered, that once you've had gene therapy, it's a one and done. But someone has asked, could a different vector or a different virus type be used to allow redosing if you were treated in the early stages, and there were discoveries following that 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 perhaps showed a more optimal treatment that might be available. So would you regret having joined in early and missing out on potentially a more effective or more positive impact from the therapy? I don't know if anyone has any thoughts on that.
So that very definitely fits into the third category of questions, I'm afraid. I think if you're taking part in these studies, you should do so in the presumption and assumption that that will be the dose of gene therapy you get. And Amy's nodding next to me in agreement. So.
Yeah, absolutely. At the moment, where we're at right now, every protocol that I have seen for a gene therapy trial has previous gene therapy as an exclusion criterion. And so I think we have to, and I'm not saying that in 20 years' time, I would sit here and probably give you a different answer, but we have to think in the time frame of your child just now. It is a very difficult call, I think it's harder with Rett syndrome, that decision, whether you participate now or you wait and maybe try and participate later. We can't predict what the next phase of trial criteria will be, and so that's something that you should discuss with the clinician at the point that you're invited to decide collectively, is this the right thing for you and your family and your child at this moment in time? And like I said, the answer might be no, actually, this isn't right for us right now. Or it might be yes, it is. But it's very difficult for us to make decisions on what might be in the future, because at the moment we don't know. And certainly for now, and for the absolute foreseeable, I can't anticipate a protocol that's going to allow you to redose with a different type of gene therapy.
I think from the perspective of Rett syndrome per se, I think the slightly different issue is that initially trofinetide, if you use trofinetide, it was an exclusion criteria, or at least was being considered an exclusion criteria. And so though it's not a gene therapy per se, but it's a biological in terms of the middle thing. So I think that is probably an important consideration to be made, openly discussed about it. And I'm not suggesting that the exclusion criteria may not change, because I think, it may change. But at this point in time, in at least early discussions, in some of the situations, it may pose a scenario, but of course it may have already changed or in the process of changing. But it is a question of the discussion is more about, there may be other scenarios coming in, and it may not just be gene therapy, but you have to have that conversation with the clinician when you're making the decision.
Great. I think that draws us to a close for the morning session now.